welcome to my channel. Today in this video I would like to discuss about the process of natural cell death, that is apoptosis. Let me tell you what is the difference between apoptosis and necrosis. Apoptosis is natural cell death, whereas necrosis is an artificial, or you can say suicidal cell death. What is the difference between apoptosis and necrosis? First point, apoptosis is a process of autonomous cellular dismantling and avoids inflammation, whereas necrosis is an accidental cell death due to environmental perturbations with release of inflammatory contents. Now, apoptosis is an active process, whereas necrosis is a passive process which occurs due to low blood flow, injury, or other disease. Now, there is equilibrium in the body between two genes, tumor suppressor gene and protooncogene. Actually, protooncogene when gets mutated, they actually forms oncogene which causes cancer. Whereas in the body, tumor suppressor gene always suppress this protooncogene so that there is no evidence of cancer in our body. Now, if we look into the cell cycle, there are basically two phases: mitosis, that means cell division, and S phase, that is DNA synthesis. In between those two phases, there is two gap phase, which is known as G1 and G2. In these two gap phases, there is control or regulators, that is called check 1 and check 2 gene and enzyme which actually control the cell cycle. If there is no such requirement of cell division, then after mitosis, cell gets restored and it is stabilized in G0 phase. Next, checkpoint kinase 1 and 2. These two checkpoint kinase are basically serine or threonine specific protein kinase encoded by check gene. Activation of this check 1 results in the initiation of the cell cycle checkpoints, cell cycle arrest, DNA repair, and cell death to prevent damaged cells from progressing through the cell cycle. They remain inactive in the absence of DNA damage, and activation occurs by dimerization and autophosphorylation. Nowadays, these checkpoints are mainly targeted by young researchers, and newer anti-cancer drugs may come which actually acts on this phase, on this enzyme, checkpoint kinase. Now coming to the main point of our discussion, apoptosis pathway. There are mainly two pathways, extrinsic and intrinsic pathway. In extrinsic pathway, the cell receives a signal to start apoptosis from other cells in the organism, whereas in intrinsic pathway, a cell receives a signal to destroy itself from one of its own genes or proteins due to detection of DNA damage. You see, there are two phases of two pathways of apoptosis. Left side you see, that is intrinsic pathway, and right side you see the extrinsic pathway. If you see right side, the NK cells and T lymphocytes, actually from there granzyme B, the serine protease, is released, which in combination with perforin, that is a pore forming protein. So perforin plus granzyme B, they actually activates caspase cascades. Now we coming to the left side, intrinsic pathway. Injury and other external factors actually helps to mutate the gene, that is DNA, which actually helps tumor suppression gene to convert to pro-apoptotic molecule, and it is regulated by BCL2 and BAX kind of regulators. I will discuss later on what is that. Now, this pro-apoptotic molecule actually activates caspase cascade 1. Now, in this caspase cascade, there are two types of caspase: initiator caspase and effector caspase. Initiator caspase first activated, which in turn activates the effector caspase, and as a result, there is a series of action which actually kind of cascade actions, which from the internal or from the external actually activates this caspase enzyme, which in turn activates the nuclease enzyme and cytoskeleton breakdown occurs. As a result, cytoplasmic body forms, which in turn produce apoptotic body. You see the down portion of the picture, and which undergoes actually phagocytosis. So like this way, natural cell death, programmed cell death occurs. Now coming to the point, what is caspase? Caspase are family of cysteine protease that cleaves protein following aspartic acid residues, which is present in cytoplasm or in nucleus, and promotes apoptosis. Now, caspase generally exists as procaspase, that means inactive, and activated by cleavage by other caspase, producing a proteolytic caspase. I have told you initiator caspase. The example are 2, 8, 9, 10, 11 and 12 subtype, and effector caspase examples are 3, 6 and 7. Now coming to the point, BCL2 and BAX. BCL2, the word comes from B cell lymphoma, which actually regulates cell death by either inhibiting, that means anti-apoptotic, or inducing apoptosis, that is pro-apoptotic. Now BAX, that is BCL2 associated X protein, that includes BAX and BAC, are two nuclear encoded proteins present in the higher eukaryotes that are able to pierce the mitochondrial outer membrane to mediate cell death by apoptosis. Now tumor suppressor gene examples are P53. Now tumor suppressor gene is basically normal gene that slows down cell division and repair DNA mistakes, or tells the cell when to die, that means when to undergo apoptosis. When it does not work properly, cell can grow out of control, and which can lead to cancer. RB gene, that is retinoblastoma gene, which actually prevent excessive cell growth by inhibiting cell cycle progression until a cell is ready to divide. Now BRCA1 and BRCA2, that is breast cancer related gene, actually when gets mutated and actually produce the oncogene. Other than this, APC-INK4 and MADR2 genes that are also related to colon cancer. Now coming to the another point, Protooncogene, that is RAS. RAS stands for rat sarcoma. It is a kind of GTP binding protein kinase, and examples are HRAS, NRAS and KRAS. HER2, that is human epidermal growth factor receptor 2, it is related with breast cancer. Cyclin D dependent kinase, that is parathyroid hormone gene. Synthesis of cyclin D is initiated during G1 and drives the G1 and S phase transition. MYC and CMYC gene. Actually MYC is a normal gene, which when gets mutated it forms CMYC, which is oncogene. Thanks for watching. Please subscribe and like and share with your friends if you like this video, and if you are having any question or any suggestion, feel free to drop your comments in the comment box. Thank you very much.